Hey Beak, so what we're going to talk about today is an introduction to adaptive immunity. Last time we talked about your innate defenses, okay? And this is your non-specific broad defenses against pathogens out there. So like your barrier defenses, your skin and your mucous membranes. You had some cellular defenses that we talked about. You have your inflammatory response. These are all things to protect you against pretty much all the pathogens. But what about when they beat those non-specific defenses? That's where adaptive immunity comes into play. Okay, now we're not going to dive too deeply into the actual interactions of the cells this section. What we're going to do is give a broad overview today, and then the next time, okay, we're going to dive into actually what's happening with these B and T cells. Okay, so, and some other things too. So here we go, an introduction to all this stuff. So in adaptive immunity, receptors provide pathogen-specific recognition. Okay, and it relies on two types of lymphocytes. You have some lymphocytes that mature in your thymus, okay, gland just above your heart. Those are called T cells. And then you have other lymphocytes that mature in your bone marrow, and those are called B cells. All right, so this short video, it goes in pretty deeply to the entire process. So I like this video, and if you want to rewind it a couple times, watch it through a few times, go ahead. And like I said, by the end of our next lesson together, where we really dive into adaptive immunity, all of this should make sense to you. But here we go.
Lymphocytes are a subgroup of white cells that make up the core of our immune system, protecting the body from an endless stream of antigens. There are three types of lymphocytes: B cells, T cells, and natural killer cells. Natural killer cells play a primary role in our innate immune system, and they are not antigen-specific, meaning they do not rely on surface antibody receptors to activate their function. We'll learn more about natural killer cells in another video. B cells and T cells, on the other hand, are antigen-specific, and in this video we'll take a closer look. Both B cells and T cells utilize surface receptors in activating their functions. These surface receptors are protein molecules commonly known as antibodies, and formally known as immunoglobulins. B cells and T cells are primarily associated with our adaptive immune system, and they work together in dynamic ways to detect and eliminate an almost endless number of antigens. B cells are found circulating in the blood, and they are also stored in the lymph system. The surface of the B cell is covered with antibody receptors, precisely matched to specific antigens. For this reason, there are a countless number of unique B cells found in our blood and lymph system, each ready to respond if it encounters an antigen that matches its own surface receptor antibodies. T cells are found in the thymus, where they are stored as immature lymphocytes. When called into action, the immature lymphocytes differentiate to match the antigens of the activating cell, and are then released into the lymph fluid and bloodstream. There are two primary types of mature T cells: cytotoxic T cells and helper T cells. Now let's take a look at how these cells work together in fighting antigens. When an antigen first penetrates the body and is detected by a matching B cell, the B cell sets off a chain reaction in the immune system. First, the B cell will ingest the antigen, break it apart, and then present a piece of the antigen onto a surface marker of the B cell. The B cell must then be activated by a helper T cell in order to begin producing antigen-specific antibodies. Other white cells, such as dendritic cells, are also involved, as they will bind to the antigen, ingest it, break it down, and then present a piece of the antigen on the surface of the dendritic cell. Meanwhile, chemical signals from the B cell flow into the lymphatic system and to the thymus, where they trigger the release of helper T cells, which migrate to the point of infection. When the helper T cell comes into contact with the dendritic cell, it will bind to the antigen-presenting marker. This activates the T cell, which then divides into both memory cells and effector cells. The memory cell is basically a copy which remains circulating in the body, ready for any future attacks by this specific antigen. The effector cells migrate to the B cell, where they bind to the marker with the matching antigen. This process performed by the helper T cell provides a safety check for the immune system to confirm the presence of an antigen before triggering the alarm. Once the helper T cell effector binds with the B cell, it activates the B cell, which will then begin dividing. And when the B cell divides, it will create both factory B cells as well as memory cells. The factory B cells produce matching antibody molecules that are released into the bloodstream, searching for and binding to that specific antigen. The memory cells are clones of the B cell, which retain the information about the specific antigen and circulate in the blood, providing defense for the future. At the point of infection, B cells with matching surface receptors, as well as free-floating antibody molecules, will bind to the antigen, helping to prevent the antigen from binding to cells in the body and thereby depriving the microbe the food it needs to produce energy to divide and multiply. White cells, known as phagocytes, also then assist in ingesting and eliminating the antigen. Cytotoxic T cells then come into play by targeting the infected human cells. By releasing toxic proteins, the cytotoxic T cell causes the ultimate destruction and death of the infected human cell. In addition, cytotoxic T cells also target defective human cells, such as cancer cells.
So, antigens are substances that can elicit a response from your B or T cells, right? And exposure to the pathogen activates these cells then that have the right antigen receptor. Okay, so here you can see mature B and T cells with these receptors, okay, and we're looking for a specific pathogen, okay, and you don't bond the entire antigen. What you do is you bond a part of it called the epitope, okay, and the B and T cells, of course, have these receptors, but each lymphocyte is specialized to recognize a specific type of molecule, okay, so, and that's pretty amazing. When you think about all the pathogens out there that we're battling, that means that we have to have that many different B and T cells ready to roll. Okay, and you might be thinking, wait a minute, if these are proteins, how do we have that many genes to match up to make all these different proteins? And that's miraculous, right? And we'll see how we do that in just a second here. So antigen recognition by B cells and antibodies. Okay, so each B cell antigen receptor is a Y-shaped molecule with two identical heavy chains and two identical light chains. There's a constant region of the chains that varies very little among B cells, whereas the variable regions, though these can change quite a bit. Okay, so nothing much happens in this area. This is pretty much always going to be the same, but this is where you're going to be different to accept those epitopes. So all these different antibodies or B cell receptors have to be a little bit different. They have to vary. So the variable regions provide antigen specificity. Right? You can see there's some disulfide bridges in these guys holding them together. And important to know, there's also a transmembrane region, okay, to these B cell receptors. So the binding of a B cell, okay, the antigen receptor to the antigen, is an early step in activating these B cells. It gives rise to cells that secrete the soluble form of the protein called an antibody or immunoglobulin or IG. Okay, and we'll see there's lots of different types of IG out there. So secreted antibodies are very similar, okay, to the receptors. However, they're not going to have that transmembrane region. So these receptors that are on the B cell, okay, are pretty much the same thing as here, only these don't need the transmembrane region because they're not receptors. They're going to be floating around. All right. So what about T cells? All right, antigen recognition by T cells. Each T cell receptor consists of two different polypeptide chains called the alpha and the beta chains. And the tips of these chains form the variable region, okay, and the rest is the constant region, similar to before, only you're not that Y-shaped, okay, and T and B cell antigen receptors are functionally different, okay, so they do work in different ways. The T cells bind to the antigen fragments displayed or presented on a host cell, okay, so the T cells are looking for you to teach them what they're looking for here, okay. These antigen fragments are bound to cell surface proteins called MHC molecules, or major histocompatibility complex molecules, and these are host proteins that display the antigen fragments on the cell's surface. All right, pretty neat. So a T cell floats around with its antigen receptors, looking for your different white blood cells to show them, hey, take a look at this little antigen I have here. Okay, this is a guy I ingested, and I'm trying to show you who the bad guy is. And if a T cell happens to match up with that, well, that can elicit the response of the T cells. let's look a little closer at that here. So in infected cells, MHC molecules bind and transport antigen fragments to the cell surface. Okay, and this is called antigen presentation. A T cell can then bind both the antigen fragment and the MHC molecule. So don't forget, these cells don't have eyes, right? They can't look and see, oh, there's an MHC out there. What are you trying to show me? This is all based on interactions, right? So when the T cell touches the antigen, it has to make sure that that antigen is being presented to it to show it, hey, here's the bad guy, theoretically, right? So what you do is you touch the antigen, okay, or the epitope fits inside your receptor, but you also have to touch the MHC and make sure that you understand, oh yeah, this is actually being shown to me from a different immune cell to teach me that this is a bad guy. Okay, so this interaction is necessary for the T cell to participate in the adaptive immune response. Okay, so B cell and T cell development. The adaptive immune system has four major characteristics. Number one, diversity of lymphocytes and receptors, right? You have so many different types of cells and receptors out there to fight all those different pathogens, like we said. You also have to have self-tolerance, right? You can't fight your own tissues, so hopefully your immune cells know enough to say, oh, you're a normal cell, we're not going to affect you right now. But sometimes that does break down. That would be an autoimmune disorder. B and T cells proliferate after activation, so there has to be a way for these lymphocytes to make clones of themselves, basically. And then, of course, you have an immunological memory. So once you fight a bad guy, hopefully you have enough memory cells around that if that bad guy shows up again, sometimes it's long and sometimes it could be decades long, right? You could have lifetime immunity against certain things. So immunological memory is very, very important. All right, let's dive into these a bit more. So generation of B and T cell diversity. By combining variable elements, the immune system assembles a diverse variety of antigen receptors, right? And those Ig, those immunoglobulins or antibodies, these genes encode one chain of the B cell receptor. Many different chains can be produced from the same gene by rearrangement of the DNA. Rearranged DNA is transcribed and translated, and the antigen receptor is formed. Oh, there we go. Okay, so see, you can actually, through recombination and deleting sections and things like that, you can actually make different receptor proteins. Okay, remember, you only have, right now we're saying probably between 20 and 30,000 genes. So how could you possibly have, thousands of different types of receptors on these white blood cells or these lymphocytes? Well, you don't have all the genes for those. Okay, but what you do is you have a bunch of genes that can kind of get moved around and mixed together in different ways to create variable regions. Okay, and hopefully you find the ones that are for these pathogens. Okay, so right now, as I'm recording this video, we're dealing with the coronavirus, right? The pandemic. And what's going on is because we've never been exposed, we don't have that immunological memory, we don't have the right receptors yet. Some people maybe do have them floating around, right? They've arranged it in a certain way and they'll get lucky. And when the coronavirus hits, okay, this type of coronavirus, the novel coronavirus, when it hits, it takes care of it. But other people don't have that yet, and thus you're going to get a little sicker. You're going to have to wait for your T cells and things like that to activate to find the actual right type of receptor. And hopefully that'll happen before too long, right? And you can get better. So there you can see how it actually works out, and you create different regions, okay, the variable region at the top. Pretty neat. Now, when I think of self-tolerance, oh boy, I get the shakes. I'm so moved by, this is amazing stuff, right? Think about this. Again, billions of years of evolution showing up here. The antigen receptors generated by random arrangement of DNA we just saw have to be checked, okay? So as lymphocytes mature in your bone marrow or the thymus or your B and T cells, they have to be tested for self-reactivity. And if these cell receptors are found that they will go ahead and attach to your own body cells, thus you could trigger an attack against your own cells, well, they'll go through apoptosis, right? They'll commit cell program death or suicide so that they can get out there, okay? And what that does is it renders these cells non-functional, and you've just saved yourself from attacking yourself. So you have this wonderful check system before you unleash these white blood cells out into your system. Hey, let's double check. It's like a quality control that's built into your immune system. So cool. So very cool. All right, so what about the proliferation of B and T cells? So in the body, there are a few lymphocytes with antigen receptors for basically any particular epitope, hopefully, right? So in the lymph nodes, an antigen is exposed to a steady stream of lymphocytes until a match is made. Remember, those lymph nodes had tons and tons of different types of cells in there, and you're just hoping that one matches up correctly. So this binding of a mature lymphocyte to an antigen initiates events that activate that lymphocyte, right? So once activated, these B and T cells will undergo tons of cell division, okay? And this is called clonal selection. So once you actually have the right cell for the specific antigen, you go through clonal selection, okay? And you have two types of cells that are going to be made. You have the effector cells, okay? They act immediately against the antigen, and then your long-lived memory cells, okay? And again, these are effectively, they give rise to effector cells if the same antigen is encountered in the future. Oh, pretty neat. So having a memory in your immune system is awesome. And this immunological memory is responsible for long-term protections against diseases. And again, it's either due to prior infection or vaccination. Okay, and vaccinations are so important, because why would you want to get whacked with a disease, potentially deadly disease, when you can just have a vaccine which tricks your immune system into learning this dangerous pathogen, even though you were never actually infected with the dangerous pathogen? Pretty cool. But of course, again, if you didn't have a vaccine and you actually were infected with it, well, then there you go. Now you still learn the bad guy. Okay, and the first exposure to a specific antigen, that represents the primary immune response. And this could take a while, okay, until your body learns it and starts to fight it off, and you might be sick for a little while, okay, or have symptoms of fighting the pathogen. During this time, selected B and T cells give rise to their effector forms. And then in the secondary immune response, so in other words, you've already fought this battle and won, and now if you get sick with that same pathogen, or I should say if you get infected with that same pathogen, okay, well now your response is going to be way faster and much more efficient, okay? A good example for me is when I was young, I got chickenpox. Now, if you're watching this video, you're probably young enough that you actually got the vaccine, okay? They didn't have a chickenpox vaccine for this herpes virus that causes chickenpox when I was a little kid, okay? And there's lots of different types of the herpes virus out there. One of them causes chickenpox. So I got chickenpox when I was a little kid. Okay, and you get these red scratchy bumps all over the place, and parents used to have like chickenpox parties and things like that because it's not all that dangerous, and they thought, you know what, if my kid's going to be itchy, I'd rather just get it over with during the summer when there's no school. Or maybe I want to get all my kids to have chickenpox at the same time so I could just deal with it. It's crazy to think like that now, but anyway, that's what happened with me when I was young. And then after I had chickenpox, I never got them again, because I have these immunological memory cells right inside of me. So when that virus hits me again, I already know how to fight it. And you actually might have the same immunity because you got the vaccine, right? So here comes the chickenpox virus, and nope, doesn't affect you. Okay, now there is something else that can happen though, because viruses, when I don't want to get too deep into this right now, but they can stay latent in your body for a while, right? So maybe in some of my nerve cells, I might still have some of the virus in there. Sometimes viruses exist as like a prophage as well, where it's just part of your DNA kind of, which is crazy. But sometimes these nerve cells can get, tricked into aggravating that virus later, and the virus shows back up, and that could be called shingles, right, when you get chickenpox later. But anyway, immunological memory, very, very important, and it's a really cool way that it's good to get viruses, or it's good to get vaccines. It's good to be exposed to different things because your immune system gets boosted. I'm friends with somebody who works developing vaccines for a drug company around here, and he always tells me that in his house, no, the rule is, sure, if food falls on the floor, yeah, there is no 5-second rule. Call it a 30-second rule, right? It's okay. It's okay for kids to get dirty and banged up, get little cuts here or there or whatever, because what it's doing is it's strengthening your immunological memory, right? The more pathogens you're exposed to, the tougher your immune system gets, and the more knowledge it has to fight in the future. Pretty cool. So we'll talk more about this soon, but here is your humoral immunity, okay, and that's like of your humors or your body fluids, and here you'll see how B cells go ahead and fight for us, which is really cool.
Humoral immunity, carried out mainly by B cells, deals with invaders in body fluids. B cells are lymphocytes that send out antibody molecules in response to certain stimuli. There are many kinds of B cells. Each kind produces and presents its own uniquely shaped antibody receptors in its plasma membrane. These unique receptors only bind to antigens with specific shapes. The B cell that successfully binds to an antigen is stimulated to proliferate into a clone of cells with the same specificity. This process of antigen-selected proliferation is known as clonal selection. B cell proliferation produces clones of plasma cells and memory cells. Memory cells are long-lived cells that can respond rapidly to future exposures to the same antigen. They account for the speed and strength of the secondary immune response that gives us long-term immunity to a disease. Plasma cells multiply rapidly. They produce and secrete antibodies in increasing amounts, peaking about 2 weeks after the activation of the original B cell. These antibodies flow through the body's fluids and tag the original foreign antigen molecules, marking them for destruction.
Okay, so again, we'll talk more about humoral immunity in our next video. Just a little intro there. And finally, let's see, can you explain this graph? What exactly is happening here? So take a look and think about what's going on. So here you can see that we're creating antibodies to whatever antigen A is. Okay, so basically at time 0 here, we got exposed to antigen A. And it took a long time, over 2 weeks, for us to build up enough antibodies, it looks like, in our blood to really fight off whatever this pathogen is, okay, that was infecting us. And eventually they come back down, and it took almost a month or sometimes over a month, okay, until, yep, the war is over, we finally beat it, we're good to go. But look what happens here. If that antigen or if that pathogen with antigen A ever comes back to see us again, look at this, whammo, in a way shorter amount of time. So here you're going from what, 29, 30 days up to like 35 days. In that short amount of time, look at how many antibodies we have here, right? And that's because of immunological memory. We have all those cells just waiting to go. Now that we already learned this virus can hurt us, we keep our receptors around, right? And you might even still have antibodies floating around in your blood here, right? They might not completely be at 0. But now you can see, oh, here's a different pathogen, okay? Because not only did the pathogen with antigen A come in here, but antigen B as well. And it's the same thing. It's your primary exposure. So that takes a while to learn it and fight it off. But now, hopefully with immunological memory, if B ever comes around again, or the pathogen that has antigen B, you're going to shoot up right away. So what we just covered today was an introduction to adaptive immunity, okay? And we didn't go too deep, but hopefully you see how the interactions happen inside of your body so that you can fight off specific foreign invaders thanks to these really cool receptors on your B and T cells, okay? And we even talked about how you don't fight your own body, hopefully, because that's a big issue, and a lot of people deal with things like that. Okay, so we'll talk more about that in the future. And also you learned about how, oh, because of this memory, as you fight a disease, you're now set, hopefully for a long time, okay, for the future when that pathogen comes around again. Okay, and when I use the word disease, this unit, I'm talking about infectious diseases, right? Not genetic or anything like that. So we're talking about little sickness makers making us sick. So again, hopefully that was helpful. And next time we'll dive in a little bit deeper to adaptive immunity. Take care.